Well, first of all, welcome to this inaugural lecture. Thank you, Kostas, for inviting me to have this honor. I'm going to try to give you an overview of the work that we have been doing during recent years, connected to the use of gold nanoparticles for a few different applications in the biomedical field. There is a risk that I will go too long, so if I go too long, please stop me. Well, let me start by showing you this very general, already a bit old slide related to what we expected from nanotechnology already for a long time. And so I don't know how old this slide is, but at least 15 to 20 years, I would say. And at that time, I think everything looked very far away. But if we look now at where all these red lines are pointing, we wouldn't be surprised to decide that there is nanotechnology, or there are nanotechnology components in many of these things, like protection coatings or lightning to advertised places, electronic menus, of course, laptops and other electronic components, smartphones. There is a lot of nanotechnology in there. There is still, however, some part which is not quite there, and this is where we are trying to advance already for a long time, which is related, for example, to health monitoring. And so we have been also speaking about personalized medicine for a long time. And we are getting closer, but I think that there is still some way to go, and we need to do more research so that we can get there. And so we have been predicting that soon we will have materials that we can plug to people's clothes or next to the skin, so that we can do sensing of biomarkers that can indicate the health level of a person, and maybe even administer drugs that will compensate whatever is not working properly. And so a little bit in this direction is where we are trying to contribute with our research. So my research actually starts about 2,000 years ago with the use of metal particles for decoration, mainly. And so I don't know if you have ever seen these pictures. These are very popular in any lecture about metal nanoparticles. For example, stained glasses in churches and cathedrals. I always ask the question, why do you think that these very bright colors have been preserved for so many centuries in cathedrals? And second, why we can only see them from the inside of the church? Any ideas? Well, the first one is quite clear, right? If we were using, or if they were using organic dyes to color the glasses, they would have been bleached a long time ago, because they are light sensitive, most of them. So they have to be made with inorganic pigments. The second is a bit trickier, and you have to know a little bit about light scattering, that if the particles that we are using to color the glass are too big, or big, then the light will not only be absorbed, it will be also more efficiently scattered. So we should be able to see the colors also from the outside. And so the answer is that here they have been using inorganic pigments of very small sizes. And the same goes for this famous Lycurgus cup, which you can see at the British Museum. And if you go to the museum and find the cup, it's not so obvious, but you can find it. You will see it's on a shelf, and there is a lamp behind it, which is switching on and off intermittently. And so you can actually see the different colors depending on whether you're looking at light transmitted through the glass of the cup or reflected from the glass. And these colors come from particles made of gold and silver, which are dispersed in glass, which would be colorless otherwise. And so this was nanotechnology. They didn't know probably, but this was nanotechnology done many centuries ago. The first scientific paper we know about these effects is from the 1850s. Michael Faraday, working at the Royal Institution in London. He published this paper called Experimental Relations of Gold and Other Metals to Light. A little bit of a scary title for a chemist or a biologist, but I would really encourage you to read this paper. It's very interesting. There's only a very small equation for optical effects. And this whole description of work that Faraday was doing in his lab, mainly related to two types of experiments. In both cases, trying to make metallic, metals as small as possible, either by etching or compressing metallic films, or by growing metal crystals in solution, so effectively making some of the first metallic colloids on purpose. And so then he basically observes with his naked eyes or with glasses, because there were no microscopes, there were no spectrometers at that time, and actually discussing the optical effects that he could observe and trying to reason the effects in the context where they still considered that the medium in between the metallic components and yourself was ether, not air, right? So anyway, a very pleasant reading, and even probably you could discover some effects that are discussed here that have been rediscovered many years after that. And so these are examples of samples that were made by Faraday at that time. At least this is what we are told at the Faraday Museum. This is one of the gold films, and you see in reflection it looks really golden, we would say. If you look very closely to the shade, it's not very clear with this lighting, and probably the picture is not the best, you will see that it looks a little bit blue. And this is because when these films are thin enough, there is a dip in the transmission, in the absorption of the film, around 550 nanometers, so basically blue light. So a little bit of blue can be transmitted through the film. If you now look at the colors of the colloids, they look completely different. And the reason is in this spectrum, which obviously was not recorded by Faraday, but where you can see that in DUV, the absorption is very similar for the film and for the colloid, but in the visible and also in the near infrared, the interaction with light is completely different. Now, what is happening here? Do we have any contamination of the metal in the colloidal state? Do we have any changes in the crystallographic lattice of the metal? Well, this is really not the case. You can see, for example, this modern high-resolution electron microscopy image of one gold nanocrystal. If you look at the crystalline parameters, the lattice constants, they are identical to what you expect for the bulk metal. So there are really no changes in the crystallography. And you can also see, if you analyze in detail, that there are only gold atoms in the lattice. So there is nothing different to bulk gold, except for the morphology and the size of the particles. And so this is where our adventure starts. And now we start talking about nanophotonics or plasmonics as a concept that allows us to manipulate light with objects that are much smaller than the wavelength of light, which is very unusual. And this is why Harry Atwater was asking whether this is a contradiction, because typically waves will like to interact with objects that have dimensions similar to the wavelength of light. For example, in opals, there is light diffraction, and this is the origin of the bright colors of opals, simply because the size of the silica particles that make up the opal is similar to the wavelength of the light with which we are illuminating, okay? So this is different in what we are looking at, because now we are looking at particles which are maximum tens of nanometers, and they are interacting with light, which is hundreds of nanometers in wavelength. And the reason for this is that in these materials, we have free electrons. And so these electrons can be easily manipulated by the electric field of an electromagnetic radiation. And so we know that in bulk metals, we can have these charge density waves traveling through the metal at a specific frequency, which is called the bulk plasma frequency, which only depends on the dielectric properties of the metal. So the frequency will be different depending on the metal that you're using. If now the dimensions of the metal are made much smaller and you have a very thin film, then these waves can only be localized at the interface between the metal and the dielectric in contact with it, because they don't fit inside of the very thin film. And then we have the so-called surface plasmons. And this is, as you probably know, the basis for the commercially available surface plasmon resonance biosensors, simply because the frequency of these waves traveling on the surface depends on the refractive index of the dielectric, the material that is in contact with the metal. And so when the bioreceptor that we have on the surface is binding the complementary from solution, the refractive index around will change. There will be a change either in the frequency or in the angle of reflection of a laser beam. And this is how we detect adsorption. Now, if we go to very small particles, so nanometer dimensions in the 3 directions, then we cannot really have these traveling, longitudinally traveling waves. So in the most extreme case of spherical particles, very small compared to the wavelength, the electric field will create a dipole on the particle. So there will be separation of charges. This separation of charges will not be at equilibrium, so they will like to come back to the uniform distribution. And there will be like balance between the force exerted by the electric field, which actually will be oscillating, and these compensation forces that try to bring electrons distributed around the particle to equilibrium. And this is where we find the so-called localized surface plasmon resonance condition. And this is which defines the wavelength or the frequency at which we will have a maximum in the absorption or extinction spectrum of the colloid. And so this is what defines that for spherical particles that are very small, a silver colloid will look yellow and a gold colloid will look red. I'll show you in a minute that if you change either the size or the morphology of the particles, these colors will not be present anymore. We will tune the resonance frequency to a different position. The color of the solution will be different. So first message of the lecture, there is nothing like a golden color. Gold can have any color, okay? So this should be removed from the dictionary. Another very important effect that takes place apart from this very high intensity in absorption is that when we are illuminating one of these particles with a certain polarization, there is the creation of a high electric field right at the surface of the particle. This is the white color that you see here. And this field will decay very quickly away from the surface. So it will be strong right at the surface, and only a few nanometers away, you will go back to the uniform electric field in the solvent. And so this is quite important because having high electric fields can affect the spectroscopy of molecules that come close to the particles. And this is one of the main bases of some of the research that we do. Now, I wanted to show you a couple of equations, but very simple ones. This is simply to reinforce that the complex dielectric function of the material really determines whether you will have plasmons at a specific wavelength or not. And if we plot these two real and imaginary parts of the dielectric function for gold and silver, we see rather strong similarities in the visible and in the near infrared. And these are that the real part is negative for both metals, and the imaginary part is positive but very small. And so this comes into a very simple equation that Gustav Mie derived in 1908 as a simplification of Maxwell's equations for spherical particles that are very small compared to the wavelength of light. And the equation looks like this. So this is the extinction coefficient. This is equivalent, so to say, to the absorbance that you measure in a spectrometer as a function of wavelength for a sphere of radius r. And so you see that there is a strong influence of the radius of the particle. So this increases a lot when you increase the volume for one particle. But it's more important to look at this part of the equation, because here we have a term which will become 0 if epsilon prime is negative, and it's minus twice the dielectric constant of the solvent around the particle. And so this will define the conditions for which you will find the maximum when you plot. All right? And this is why it's important that for silver and gold, there is this negative real part of the dielectric function in the visible or in the near infrared. And the second part is that this other term will become extremely small if you squared it, because it's already a very small number for the normal value. Okay? So this is also another interesting message, and this is why you could also use spherical gold or silver particles as a sensor, exactly the same as the thin film, because if you change the refractive index close to the surface, you will have a change in this term, and so the wavelength at which epsilon prime will become minus twice epsilon m will be different. There will be a shift in the position of the maximum. Okay? Right. Now, if you are not using spherical particles, which is usually the case nowadays, then life is not so easy, and you have to resolve Maxwell's equations completely, and these are complex differential equations, right? And so you have to go and use numerical models, and there are quite a few around. Nowadays, this is also an old slide, maybe 10 or 12 years old. Nowadays, you can really buy software packages where you can directly use these numerical methods on almost any computer. Still, if you want to calculate complicated structures, it may take a while until you get a convergent result. But anyway, you can really, much more easily than before, you can really obtain the predictions that you need for your work. I would also recommend, if you're interested in these simulation processes, to read this other paper. This was actually the work of Renwen Yu and his PhD advisor, Javier García de Abajo at ICFO in Barcelona. And they asked me to contribute by basically discussing and providing experimental examples. But what they have done is actually to develop an analytical method that can allow you to resolve the Maxwell's equations for any morphology by taking parameters from numerical calculation. So you run the numerical calculation once for that geometry, you extract some parameters, and then you can use an analytical method. If you're too lazy, simply, well, I forgot to put the reference here, but you can visit García de Abajo's webpage, and you will find widgets that you can use directly, obtain the spectrum, and even download the data. So that makes life a lot easier. And the calculation for this takes milliseconds. So then it's a lot easier. But anyway, so again, my message from this couple of slides is that if we know what we want to use for our application, we can predict it by using numerical methods or theoretical methods. You can even do it for more complicated structures if you have good friends with large computation capabilities. And this is what we found actually at the University of Vigo, where I used to work 8 years ago. There is a group of telecommunications engineers who actually tuned the resolution of Maxwell's equations because they were working on the interaction of radar waves with either tanks or submarines or whatever. And so they really needed to optimize the calculation of the interaction of these long waves with big objects. But they apparently have competitions who is able to resolve the problem with the largest number of unknowns. And they get to millions of unknowns to be resolved in a single computational run. And so for us, the concept was actually very similar, because all you have to do is now to reduce the wavelength of your light and to reduce the dimensions of your object. But now you can calculate even at the full detail. For example, we know that our gold nanorods have octagonal cross-section, so they can really draw exactly the same morphology and then calculate, not only for one particle, but actually for thousands of particles with the size distribution that we have experimentally. And so they can reproduce almost exactly the spectra that we measure. Not only that, you can also calculate groups of particles. And this is very important because when the particles are very close to each other, then the behavior is different to when they are isolated. There is something that we call sometimes plasmon coupling or plasmon hybridization. And then you obtain new plasmon modes that, for example, can create very large electric fields at gaps between particles. And this is, again, very important for our applications. And so, for example, in the context of our work, where we try to make the particles, to play with their surface chemistry, to direct their assembly into well-defined structures, and to obtain the optimal structures to improve the detection of different types of molecules, then we need to know really which is the best configuration for the detection. And so we challenge them, for example, to tell us, oh, sorry, I'm going too fast. Anyway, let's start from making particles. And you have seen already one spherical particle before. This is a collection of particles that are made quite monodispersed. This is quite easy to do. I used to do it in my second year undergraduate courses in Vigo. So you simply ask students to dissolve gold chloride in water, to heat it to boiling, and add a little bit of sodium citrate, and they obtain particles like this. And with very well-defined spectrum and with high uniformity. If you want to play around with the morphology, then it becomes a bit more difficult. You have to understand quite a few concepts if you really want to manipulate an isotropic crystalline growth at the nanoscale. But by playing around with the conditions and prior knowledge, now we are able to really make different morphologies with very high detail, very high monodispersity, and almost without any particles which have a different morphology within a single synthetic run. And the advantage of being able to manipulate the morphology is that now we can select at which wavelength we can work, and also we can manipulate the spots and the intensity of the electric fields that we can create at different parts of the particles. So these pictures that you see here are so colorful, except for this one, they are actually all experimental data that are measured with an electron microscope by doing electron energy loss spectroscopy. And these bright spots that you see at different parts of the particles are equivalent to the enhancement of the electric field that we would obtain by illuminating with light. And so now we can really do also single particle or even multiple particle analysis of the optical morphology to enhance electric fields if we want to do applications in that respect. I don't have time to go through the synthetic details. Let me tell you that once you know how to do it, it's actually quite easy. If you really want to understand the mechanisms, then it becomes very difficult because there are a number of parameters that actually influence how the crystal grows in the nanoscale, and also a number of parameters that decide how stable your particles will be. So this would take a bit longer. Now, the state of the art for gold nanorods is something like this. So you can make really large amounts of material in a short synthetic process with a very high quality. So let's say that the standard monodispersity nowadays is around 10% in terms of ratio between the length and the width, which is what in the end defines the optical response. If you go to this paper, you will find that by manipulating pre-made colloids, you can go down to 2% monodispersity, so actually to very narrow absorption bands. Again, I don't have time to explain that today. Let me just show you that by playing around with the synthetic parameters, we can tune this ratio between length and width, and this can actually decide where the maximum absorption will be, which will be the color of the dispersion, all right? And this is very well known. Many groups in the world can do something like this. So you simply go to the literature, find which are the experimental conditions for a specific aspect ratio, and then typically you should be able to get something close to that. And this is again another paper that is recommended if you want to enter the field of gold nanorod synthesis, because here we took the effort to disclose all the tips that we know, what is important to take into account so that you can make high quality gold nanorods. All right, now going into applications, as I thought I'm going a bit slow. 20 minutes, that's well, okay. Right, so let me tell you a little bit about sensing. Raman scattering is one of our favorite techniques, mainly because it is very highly selective. And so just as a quick reminder, in the degree of chemistry at least, when you study Raman scattering, you do it in close connection to infrared spectroscopy, mainly because both techniques are related to vibrational transitions. But they are very different, because in infrared, you're using infrared light to excite vibrations of chemical bonds, and then depending on the frequency that is absorbed, you will know which chemical bond you're exciting. In Raman, you're exciting electrons to a forbidden state typically, and then you look at scattered light from the emission of the electron that goes back to the same electronic ground state, but sometimes to a different vibrational state. And then this difference in the scattered radiation compared to the incident radiation will tell you which is the vibrational level that you're exciting. The main advantage of Raman is that there is no contamination in the signal from water. So it's very easy to analyze aqueous dispersion. The disadvantage is that because we have a forbidden state involved in the process, the efficiency is extremely low. Or in other words, the probability of a Raman transition is very low. And this is why it has rarely been used as a highly sensitive analytical tool. However, it was discovered in the 1970s that if you're looking at molecules that are in contact with, in that case, graphened metallic electrodes, nowadays we would say nanostructured metallic systems, then you can obtain the same spectra, but amplified by many orders of magnitude. And the reason behind that is, as I already mentioned earlier, you are creating a very high electric field, which will affect the polarizability of the molecules if they are close enough to the surface. And then the probability of the Raman scattering transition will be enhanced a lot. In theory, you can go up to enhancement factors of 12 orders of magnitude, but in order to get those, you have to optimize your substrate and your process. And so, for example, you can manipulate the morphology of the particles, as I showed you, so that you obtain the largest electric fields. But pay attention that in these cases, the largest electric fields are confined to very small volumes. So if you want to really exploit this very high electric field in a nanostar, you need to bring your molecule in contact with the tip. If the molecule goes to the center, it will not feel a very high electric field. Okay? Another way to increase the electric field enhancement is to bring particles close to each other. I also showed you before, and this is a very clear example. This is actually simply a gold colloid with spherical particles, quite dilute, and then we introduce organic molecules that are Raman active. If we measure the colloid in solution, the enhancement that we get is not enough to amplify the signal of the concentration of molecules that we had. But if you simply add sodium chloride to the solution, drive the aggregation of the particles, many molecules will be located at gaps between particles, and then immediately you get a spectrum like this, where you can identify basically all the vibrational peaks corresponding to those molecules. So simply by bringing particles close to each other, you get a much larger enhancement of the Raman signal. The problem is, if you do it that way, that it will be random. And so you will never know exactly how much you're amplifying at each part of the aggregates. And this is why I have my own list of advantages and drawbacks. that, in my opinion, are the major reasons why surface-enhanced Raman scattering is not yet in the clinic or in the market. And so there are many reasons why it should be, and I already mentioned a few, but there are also reasons why it is not, and they are related to the reproducibility of the substrates, the uniformity within the same substrate. As I said, if you're aggregating particles on top of substrate, the organization of the particles, it can be completely different at different spots, and then the enhancement will be completely different. Another point is that the analyte has to be very close to the metallic surface. And so sometimes we were saying the analysis is restricted to molecules that have some chemical affinity for a metallic surface. Nowadays, we have tricks to help solve that problem, but it's still a problem that has to be solved. Another point is, especially if you're going to bioapplications, in biofluids there are plenty of other molecules around. So if you're interested to study, for example, a cancer biomarker, you need to find really the peaks which are specific to that marker. And if you have other proteins or lipids floating around and they get in contact with your metallic surface, they will, on one hand, bring many more vibrational peaks to the spectrum, so you will have a hard time identifying the one you're trying to identify. On the other hand, they may actually block the surface, so your analyte may not be able to reach the contact with the metal. So again, for those who are interested in this technique, this is a major review that we just published. It's actually written by most of the major players in the field. I think we are like 65 authors or something like that. But as you can see, it's a long one. So it really covers most of the aspects that you can really think of in the current state of the art of the technique. All right. Again, going back to my colleagues in Vigo, I was going too fast, and I was saying we challenged them. We challenged them to compare what we should obtain if we are bringing gold nanorods close to each other. But we are actually creating like an extended film of nanorods, and they can be either completely disordered, randomly ordered on a film, or compare with a stack of crystalline arrangement of nanorods which are standing up on a substrate. And you may say, why do we think of this? Well, we think of this because we think that the interaction between particles will be more efficient if they are well ordered. Second, because we have a hint that we may be able to synthesize something like this. Anyway, so the response is that with the randomly oriented film, you very quickly lose the enhancement because the light gets absorbed. There are no constructive interferences, and only a few tens of nanometers below the surface, you will get a dark map if you calculate the enhancement factor. In the crystalline arrangement, every plane in between two monolayers of nanorods will give you something like this, so you have a lot of hotspots that are active for the detection. So now the synthetic challenge is not only to have highly ordered arrays of nanorods, it's also to allow the analyte to diffuse in between the monolayers. And so this actually came in the context of one of, so my first year C grant, which was related to the design of substrates for the detection of communication between bacteria. I'll show you more in a minute. And so I recruited Cyril Lamont, who had been a student in France, in Rennes, and he actually had a technique to grow these oriented crystals. And so because we wanted to have a high uniformity to be able to detect the same thing at different spots on the surface, I asked Cyril to try to create these crystals within the holes of a PDMS stamp. So you simply have a droplet of the gold nanorod colloid, you bring a PDMS stamp with holes, you put it on top, and you wait for the water to evaporate through the tiny pores of PDMS. And after some optimization, he was able to create this sort of substrates with many towers where if you look inside of each of them, you find that there is really a crystalline arrangement of the nanorods. Now, in order to create a path for small molecules to diffuse in between the monolayers, we simply incubated these substrates in a sol gel solution with surfactant, so that we created a mesoporous film that would actually infiltrate in between the monolayers. This was quite lucky, I should say. But by doing that, we were really in a position to study some metabolites which derive from the communication between bacteria. So very quickly, the way this works is that in order for bacteria to create a biofilm, for example, they have to know how many individuals are present in the community. And they know that by releasing some molecules, very often homoserine lactones, which are selectively recognized by some membrane proteins. And when this happens, the genetic cycle of the bacterium will change, and they will start doing different things. And so it's important to have a method that allows you to read that communication in a non-destructive manner. And so what we did in the end was to have these pillars of oriented nanorods covered with silicon oxide and infiltrated with a mesoporous film. And then we actually grew bacteria on top, and we started to see the signal of this molecule, pyocyanin, at very early stages during the growth of the bacteria, so we could actually see when they start to create these biofilms at some point. In fact, we also did a similar piece of work with hydrogels that could be implanted below the skin of a mouse. In this case, this is a dead mouse. But what we wanted to see is whether we can actually do the detection through the skin. And we were able to identify sub-micromolar concentration of the molecule when it was implanted below the skin of the mouse. So this is why we are proposing that this might be useful to monitor infections, for example, after surgery. We have a lot more work that I cannot show today, for example, related to looking at interactions between different bacteria families. And so we look at different biomarkers for each of the families, and we see how one type of bacteria can actually inhibit the production of the other biomarker in the other bacteria type. And so this also can help you learn how they can fight for certain whatever food or other propagation effects in different conditions. Now, I have to go a bit quicker. Now we actually improved the fabrication of these substrates. This is a collaboration with Agustin Mi at ICMAP, again in Barcelona, who can make nanostructured templates with much smaller features. And so now we can play the same trick as before, so simply using the stamp. But now we can create these very well-organized patterned arrays where the number of particles in each spot is much smaller. So again, much easier to understand. But more important, the distance between each of the aggregates is in the order of a few hundreds of nanometers. So it can create diffraction of light. And so when we have this, we have something called lattice plasmon mode, where the absorption is defined mainly by the spacing between the clusters, and it creates a much larger enhancement of the electric field at each of the clusters. And so this actually gives more sensitive detection. And now we are using it already in the second year C, which is related to applying similar concepts, but to study the behavior of tumors. And so we are starting by studying in vitro the evolution of some biomarkers that are related to cancer cell activity. So for example, if you study cancer cells that overexpress the IDO enzyme, you can actually watch the consumption of tryptophan and the synthesis of kynurenine. And so my student was basically analyzing the peaks of each of these two bioanalytes, which you can rather easily detect in these complex fluids. And then he basically fabricated a microfluidic chip where he can grow the cancer cells in contact with extracellular matrix. One of the windows now will be the nanostructured substrate. And then by overexpressing the production of the enzyme by simply using interferon gamma, then in real time we can watch the growth of the peak of kynurenine and the drop in the peak of tryptophan. So we are expanding this study to other tumor processes. And more important, we are trying now to implement similar enhancing systems in three-dimensional substrates, so we can actually grow tumors in 3D and try to observe what happens to them at different parts of the tumor. Within the same context, we also wanted to be able to label cancer cells and monitor them in real time over extended periods of time. So typically you would do this by using, for example, fluorescently marked antibodies, for example. We are using something that has been proposed a long time ago, which is actually using single particles, which are very efficient at enhancing Raman signal, as labels or as fingerprints or as barcodes to monitor the position of cells. So nanostars are one of our favorite materials for this. You have already seen that they can really enhance the electric field a lot in their tips. And so by tuning the morphology of the nanostars, you can actually arrive at a maximum enhancement around 800 nanometers, which is already within the first biological transparency window. And now what you can do is attach what we call Raman reporters, which are simply molecules with very well-defined Raman scattering signals, and then cover them with an inert material, and then use these coated particles as tags that can be bound to cells and then monitored over long periods of time, because as opposed to fluorescent dyes, these signals do not bleach. Okay? So the way this is simply to start from a dispersion of gold nanostars. Then you cover them with a thiolated, typically aromatic molecule. Then these particles become hydrophobic, and then they can be dispersed, for example, in chloroform. And then they can be wrapped by a block copolymer. This is a technique that Wolfgang Parak and other people developed already a long time ago. And it works beautifully. So you can simply change the nature of the molecule that you have inside in order to obtain different fingerprints. And then with the same block copolymer, you obtain something which again becomes hydrophilic and can be functionalized because you have a lot of OH groups here on the outer surface. And so we selected, for example, in this case, 6 different organic molecules. So we have 6 different codes that can be internalized in different cell lines, and then you can simply identify the presence of the particles inside of the cells by doing Raman mapping. Now you can also mix the cells and have a co-culture of, for example, 5 or 6 different cell lines, and then let me go quickly through this because it's late. Then you can very quickly do a scan by simply looking for peaks which are characteristic of each of the codes that you have in different cell lines, and you can really identify the position of the different cell types within a planar 2D culture. And this can be done over long periods of time, as I said. Now, you can also functionalize the particles on the outside. You can, for example, label again with a fluorescent dye. So for the initial steps, you can also do fluorescence labeling. And because these particles also reflect a lot of light, you can do dark field optical microscopy and use it as a complementary technique. In another collaborative work where the group of Costas also participated, our joint student Rafaela was working to demonstrate that these gold nanostars are also very efficient for photoacoustic imaging. So we have another additional technique that can be used in combination with the same material. The way we are going at the moment is again into 3D, and we just got another paper accepted where we show that we can very clearly do imaging of multi-layer cell stacks by labeling different cell types with different nanoparticles and doing the imaging in 3D, so looking at interdigitation, for example, of cell layers with each other. And so these are sort of the first steps to go into more extensive 3D characterization by using these combined surface-enhanced Raman scattering techniques. Now, if I still have 5 minutes, do I have 5 minutes? I would like to tell you something which is, in a way, completely different. And it's the application of a different plasmonic effect for, again, biosensing, or at least for monitoring biological processes. This is also related to the coupling of plasmon modes. And well, in this perspective article, you can read a variety of possibilities for optical effects that arise from organizing particles in different ways. So this was the easy one I showed you earlier, where two particles come close to each other, and then you get the plasmon hybridization. But think, for example, if you're able to organize particles in a helical structure, then you will obtain a chiral object. And the question is, will then light couple with these particles that are organized in a chiral arrangement with optical activity? Will you be able to obtain circular dichroism in the visible or in the near infrared? And of course the answer is yes, otherwise I wouldn't include this in the talk. And there have been quite a few examples in the literature, mainly based on using DNA nanostructures as templates. This is the most common way. Either using simple DNA strands, or most commonly using, for example, DNA origami, where you can position very carefully functional groups where the gold nanoparticles will attach. So you can see, for example, create a helical DNA origami structure where gold nanoparticles will be placed following this helical orientation. Or you can create DNA origami sheets where gold nanorods will be attached on a specific angle. And so you can actually rotate in different planes the orientation of the nanorods. And this will give you a very efficient chiral plasmonic structure. However, if you want to go into real applications, these strategies are a bit difficult to implement. So in an older piece of work, almost 10 years ago, Andres Guerrero, who was a postdoc in my group, he's now a professor in Madrid, he actually managed to obtain chiral fibers, organic fibers, that form a gel into helical fibers. And the helicity, so the handedness of the fibers, was defined by the chirality of the monomer molecule. And by doing that, he found that if you're able to attach gold nanorods following the helicity of the fibers, even if it's not perfectly well organized, it will give you a very strong circular dichroism signal around the position of the plasmon of the single nanorod. And so this offers already more possibilities for practical application. And so when I was already in San Sebastian, there was an application from an Indian postdoc who wanted to apply for a Marie Curie fellowship. And he wanted to do something related to optical activity, but his background was more related to conjugated polymers. And because we are in a biomaterials institute, I said, well, I think that going for conjugated polymers is not the best idea for us. Let's try to propose something which is more related to the biomedical field. And then I remember some discussions that we had related to the similarity of these fibers that Andres was using with amyloid fibers that are responsible for many neurodegenerative disorders. And so the proposal was related to studying the possibility of monitoring the aggregation of amyloid proteins into helical fibers. And so we mainly focused on, well, of course, the grant was approved, and so Jatish came and started working. So initially he started working with Aichiber Kortajarena, who's a group leader in our institute, an expert in protein expression and also in the fabrication of nanostructured materials from proteins. And so she was able to express the protein alpha-synuclein, which is a model protein for Parkinson's disease. And so this was expressed from bacteria and then incubated under the conditions that it will aggregate into fibers. From the literature, we knew that most of these fibers can have this double helical structure, so they would be really ideal templates if we were able to absorb gold nanorods that follow the helical structure. Now, one issue was that the dimensions of these fibers are a little bit small for the typical nanorods that we make. And so the first challenge was actually making smaller rods that would fit within the pitch of this helical structure. And so Yatish actually was able to make rods which were somewhere 7 nanometers in diameter and maybe 20 nanometer long that would somehow be able to fit following this helical pitch. After doing that, he was simply extracting samples from the incubation of the protein at different times and then mixing with the gold nanorods, and after equilibration measuring the circular dichroism signal. And this is a summary of what happens in an optimal case, where you can see that the intensity of the CD in both sides is actually increasing for samples that have been incubated for longer times. If you now take samples from these different times and go to the electron microscope, you can see pictures like this, where longer and longer arrays of gold nanorods are being obtained. If you are a true believer, like I am, then you can very easily see some places where the rods seem to be crossing each other, as you would expect for a double helix. If you are a scientist, as I also try to be, you really want to confirm that this is what really happens. And then you have to go and ask your friends to give you a hand because you don't have a proper cryo-electron microscopy service. And so we went to our colleagues at the University of Antwerp and the group of Professor Sarah Bals. They are extremely good at doing electron tomography. It was a little bit challenging for them to combine this with cryo-electron tomography. But in the end, they provided us with a few movies like this, where we really identified something very close to a double helix. And in fact, because this is coming from a real measurement, they can provide the coordinates of every spot in the nanoparticle. So we can actually now go to our other friends who can do the simulation of anything. And then they actually simulated the circular dichroism from this specific structure. That one was very noisy, so it's in the supporting information of the paper. The ones that we show, oops, it's not even here. I thought I had the simulation. Anyway, so for very well-organized structures, you really obtain something very similar to what we measure experimentally, especially if you take into account the polydispersity in the organization of the nanorods. Now, in order to show more realistic application and not claiming that we are going to cure or diagnose Parkinson's for the moment, we went to our colleagues at CSC BioGune